We finish these lectures on immunity talking about T lymphocytes. And T lymphocytes are involved in cell-mediated immunity. They regulate the immune system. And they also deal with cells that have gone off the rails. The way they recognize antigens is using T cell receptors, or TCR. These are similar in structure to antibodies. Or you have these two protein chains that combine. They have a variable region and they have a constant region, as shown here. Here's a cartoon of what it looks like, and here's actually a space-filling model of alpha and beta. However, unlike B cells that can respond to antigen directly, sometimes, T cells respond to antigen only when association with another host cell. The antigen-presenting cells place the antigen in a major histocompatibility complex molecule and then present it to the T cell. So let's spend some time talking about MHC molecules. Almost all human tissues contain MHC molecules, and there's two types. MHC1 is found on almost all cells. They are status monitors for cells. MHC2 are found only on antigen-presenting cells. Again, macrophages, dendritic cells, and B cells. Here is a cartoon of an a MHC molecule, and you can see that you have again a structure composed of 3 or 4 proteins here, and then there's a groove where this antigen is presented. Here is a top-down space-filling model, and here is a side view showing the MHC molecule, and then also showing an antigen in the groove of this, and this is presented to the T cell. So MHC1 function. It binds to peptides that originate inside the cell. As a normal course of the processing of cell, some of the intercellular proteins will be digested by a proteasome, and they'll make these little peptides. These peptides are then incorporated when a MHC1 molecule, as the MHC1 molecule is synthesized, and they eventually go through the endoplasmic reticulum, the Golgi apparatus, and they end up on the surface of the cell. If a cell is healthy and normal and nothing's going on inside, the MHC1 molecules will only present self-antigens. If, however, an intracellular parasite, i.e. a virus, is present in the cell, some of its proteins will be digested, and these viral proteins will be displayed on the MHC1 molecules. If that happens, the immune system will detect this and react. So it's a way of signaling whether a cell is infected or not. MHC2 molecules are different. These are present on cells of the immune system, and they serve as vessels to present foreign antigens to T cells. So when let's say a macrophage comes in and destroys a pathogen, so this extracellular pathogen comes in, it's destroyed, the little pieces of it are digested, they are put on synthesized MHC2 molecules, and then this is presented to the immune system. Basically, what you're presenting is leftovers of the phagolysosome, and these and presented to T cells. Dendritic cells can also do this, but instead of killing pathogens, they just pick up material that's in this body somewhere and then process it and present it to T cells. There's 2 types of T cells. There are CD4 plus cells. These cells are involved in regulating the immune system, and there are CD8 cells, which are cytotoxic T cells. CD4 plus cells can be further divided into 3 groups. Th1, which are involved in cell-mediated immunity, and they recruit and regulate other cells. And Th2, they are involved in antibody production. They regulate B cells and antibody production, and they're involved in antibody-mediated immunity. And then finally, T suppressor cells that turn down the immune response after a pathogen is dealt with. CD8 cells, again, are cytotoxic T cells. They are activated by an antigen, and this will cause these CD8 plus T cells to attack the cell displaying it. And they can be thought of as sentries in your body. Here's a comparison of the reactions of CD8 and CD4 cells. On the top, you have a target cell or an antigen presenting cell. So a cytotoxic T cell, a CD8 cell, it will go around the body and it will interface with these MHC1 molecules and check the antigens that are there. If they're self-free antigens that they don't react to, they'll just move on and the cell is left alone. If, on the other hand, this is a non-self protein that this T cell receptor recognizes, it will activate the T cell. Antigen presenting cells, again, B cells, macrophages, and dendritic cells, process antigen. They presented in MHC class II molecules, and this is presented to CD4 plus cells that then go on and regulate the rest of the immune system. Now, one of the questions you might have is, wait a minute, you've been talking about how the immune system can recognize self versus non-self. How is that actually manifested? How does that happen? Well, what happens is a T cell, after it is made in the bone marrow, migrates to the thymus. These T cells, when they're made, are programmed for cell death. If nothing is done, they will go through apoptosis and die. When it comes into the thymus, it's then educated. It's exposed to various tests. The first test is to see if it actually has good signaling or receptors. If it does, it's allowed to live. If not, its cell programming causes it to die. It's then tested to see if the T cell receptor can interact with an MHC molecule. Again, if the interaction isn't productive, then the T cell is again allowed to die. Then finally, you test this T cell against various self-antigens present in the thymus, and if any of them react to the self-antigens, again, it's an autoreactive T cell, it's allowed to die. Otherwise, its programming is changed, it's stabilized, and you now have a mature T cell that can migrate out of the thymus and then to various parts of the body. 90% of the T cells that are made end up dying because of this education process. B cells aren't culled the same way, but they only activate well with the help from T cells and T suppressor cells that can control inappropriate responses. There are a number of other tolerance mechanisms. In clonal anergy, a cytotoxic T cell may react to a self-antigen when presented in an MHC1 molecule, but its partner Th1 helper cell will not react in the same way to the self-antigen, and thus the cytotoxic T cell is never activated. Secondly, T suppressor cells will shut off normal immune response. If there's a self-reactive T helper cell, there's often a companion T suppressor cell that will shut it down. So that shuts down apparent responses right away. Finally, there's sequestering. For example, the cornea in our eyes has no vessels and no blood vessels, so there's no access to the immune system to it. That's why you can transplant cornea between people without a problem. Okay, a 3-minute paper. What I want you to do is take 3 minutes and explain this question. How do we detect self from non-self? In your explanation, use antigen, antibody, T cell receptor, dendritic cell, and thymus. Okay, hopefully you incorporated the stuff that we had just talked about. T cell activation. T cell activation follows a similar pathway to B cells. Again, you can see that in the textbook in chapter 16. An antigen presenting cell presents an antigen. Several signals are then necessary to activate a T cell. There must be a complex between the MHC2 molecule and the T cell receptor. Secondly, a signal between B7-2 and another membrane protein on the antigenic receptor and the CD28 receptor must occur. When these two things happen, you get a response. When a cytotoxic T cell is activated, it first recognizes an antigen. It then is reinforced by a Th2 helper cell and activated. When it recognizes that antigen for a second time, it kills. And cytotoxic T cells kill in two ways. First, the TC cells have FAS and tumor necrosis factor ligands on their surfaces that bind to FAS and TNF receptors on the surface of the target cell. This precipitates a signaling pathway that leads to apoptosis or cell death. So it turns on switches on the cells that causes them to go through apoptosis, cell death. Secondly, fully differentiated T cells have granules inside them that contain perforin and gramzymes. The T cell makes contact with the target cell and then releases the perforin. This polymerizes and forms pores in the target cell membrane, allowing granzymes to then release from the T cell and migrate through the perforin pores into the target cell. You've now punched holes in these cells that are under attack, and then the granzymes, or serine proteases, they activate nucleases and caspases in the target cell, further encouraging apoptosis. The combination of these two reactions will kill the target cell. And remember, this target cell is under attack because it contains foreign antigens. For example, if a virus is infecting this cell, the T cell will come by and kill that cell, thus eliminating the virus. Okay, a top hat question. What is true about T-C cell interactions with MHC? The correct answer is A. TC cells recognize antigens in the context of MHC1 molecules. So all of this involves T helper cells. If you go and you look, these T helper cells, these CD4 plus cells, will regulate the immune system. They promote tumor immunity, and they get rid of intercellular pathogens, Th1 cells, and cell-mediated immunity. And Th2 cells help in producing antibodies. So these T helper cells are very important in dealing with the immune system. Okay, finally, and the last thing, we're going to put it all together. Please go and read section 16.5 of the textbook to get an idea of the entire process. This goes through this in high detail, and I don't want to do it here. You can do it there. Okay, but what it describes an infection by Streptococcus pyogenes. Once you're done with that, please do a Dottie defense mini-map. Imagine the invasion of Streptococcus pyogenes, which is talked about in section 16.5. Go through and order all the different things that happen and what order they happen in. Okay, that will be really helpful for you to understand this process. So here's the answer to the body defenses mini-map. Some of these may be slightly out of order in your sequence, and that's probably okay. For example, complement activation and phagocytosis might be switched, but you should generally have this sequence. Now read the next section of 16.5 that describes a viral infection. Note a very similar response would happen to COVID-19. Your immune response is amazingly powerful and can defend you against pathogens that haven't even existed yet. The current COVID-19 epidemic is a prime example of this. While too many people will die from this infection, realize that at least 90% of individuals that get infected will not. This is a virus to which we are immunologically naive. We've never seen it before, yet most of us are able to defend ourselves against it. The immune system response is multiphasic, with innate response first defending against the pathogen. If the innate response fails, then the adaptive response is called an reaction. Once you have been exposed to a pathogen and survived, in most cases you are protected against that because of your immunity. And this immunity is expressed in the sense of memory B cells and memory T cells. you have an amazingly powerful immune system.